Perfect.
Okay.
Yes, because
Okay.
so you can see my slide, right?
Yes.
Perfect. So it's a real pleasure for me to be here today, guys. So thank you for the invitation. As Rolando already said my title, I am not repeating it, but this talk is about, or I am going to show you how to apply a network model to both highly exploited benthic ecosystem in Chile. And I would like to start to say that this work is in collaboration with Pablo Marquet and Sergio Navarrete from the Pontificia Universidad Católica de Chile, and with Fernanda Valdovinos from UC Davis, and who were, they three were my PhD advisors during my PhD thesis. So this talk or the work that I am doing is under the umbrella of overharvesting by fisheries. And why? Because overharvesting by fisheries is a global problem that is threatening marine ecosystem. In fact, this is one of the main causes of biodiversity loss in coastal ecosystem. And why this is important and why is interest for me, because when we think in fisheries, we know that fisheries directly have impact of the target species because decrease directly their biomass, right? And to fishery management, they are interested to understand all the factors that affect the biomass of these target species. But we also know that fisheries could have impact on the rest of the ecosystem, because as you can see in this picture, the species are not alone, they coexist and interact with several other species. So if we could map all these interactions, start to emerge this complex network of interaction. In this case, this is the intertidal network of interaction of the benthic coastal ecosystem of Chile. Each species or each node is an species and each link is an atrophic interaction. So here I am representing in this node a target species. So now if we think in fisheries, we know that if fisheries decrease the biomass of a target species, this impact could propagate through the networks and could produce biomass changes in the biomass of the other interacting species, right? And this could also produce impact back to this target species, and maybe could also produce undesired impact on the other harvested or not non-harvested species in the ecosystem. And I know that if you hear that sound quite obvious, if we compare the number of studies that assess the impact of fisheries at population level, this is in the target species, or at community, food web, or network level, clearly here we have the lower proportion of studies. And what this means that we know very little about how the fisheries impact propagate through the food web and how could affect the ecosystem. So based on that, I am interested in understanding the impact of fisheries on food web using multi-species approaches. So according to study this, I use these two study systems, the shallow subtidal and the intertidal rocky shore of central Chile. These two ecosystems represent all the trophic interaction that you can find along 700 kilometers of coastline. And these are the type of species that this food web represents, from plankton, benthic diatoms, we have some macroalgae, filter feeders, we have a very important carnivore here, which is loco, the Chilean abalone, which is have a very high importance in terms of ecology importance and in terms of economical importance. In the subtidal network, we also have some fishes and sea urchins. And in the intertidal food web, we also have birds. And the type of species that have these red boxes represent the type of species that are being harvested by fishers. So what this means that fishers artisanal fisheries in Chile is a multi-species fisheries. It's removing several species at the same time. And the type of fishery that is that we are studying is the artisanal fisheries. Why? Because these shallow subtidal and intertidal ecosystem are located in this coastline. So in Chile, we have this law that the first 5 miles of coastline are exclusive for artisanal fisheries. So this is a reason of why we are working with artisanal fisheries and not with industrial fisheries. So these are the networks. This is the shallow subtidal, and in the right, the rocky shore intertidal. Again, each node represents a species, and each link represents an atrophic interaction. So both food web have more than 100 species and more than 1,000 of interactions. And the species are organized from the bottom to the top, with primary producers as macroalgae and top predators in the top, in which the letter F represents the fisheries node. And if you see the nodes, they have 2 colors. The red ones represent the harvested nodes, the harvested species, and the yellow ones represent the non-harvested species. And also, if you see the nodes, they have different sizes. And the size of the node is associated with the number of interactions at which that node is associated. So just looking this, we could say that fisheries is removing a species along the entire food web, right? Because it's removing at the higher trophic level and in the base of the trophic level, the lower trophic level. But also, these red nodes are these biggest red nodes. And this means that fisheries is removing highly connected species. So just looking this, we could say that the impact of fisheries could propagate through the entire network. So according to understand how the impact of fisheries propagates through the food web, we simulate the food web dynamic using this allometric trophic network model, which is basically a set of these 2 equations, one equation for each basal species and one equation for each consumer. And sorry, let me hear. Okay, so and the basal both equation is a balance between the biomass gains and the biomass losses. And in the case of basal species, they could gain biomass through growth gain, which is a logistic equation in which the carrying capacity, this K, is the ecosystem carrying capacity. And what this means? Means that all basal species compete to reach that carrying capacity. And that interspecific competition is represented by this parameter C. And basal species could lose biomass through predation loss or through fisheries loss. And this fisheries loss we are modeling as the simplest way that we can model fisheries as an exploitation rate, which is the Fmax that directly depends on the biomass of the target species. So this Fmax could take value from 0 to 1. 0 means no fisheries, and 1 means that fisheries remove the 100% of the biomass that is available of each target species. And in the case of the consumers, they could gain biomass through resources consumption and lose biomass through maintenance, through predation, or through fisheries. And in the case of the consumer, this F parameter is the functional response, This is equation in which is this functional response is a mix or a combination between the type 2 and type 3 functional response, which are defined by this Q parameter. Okay, and in our network, we have the plankton and the detritus node, which we know that the dynamic of these two nodes do not depend very much about what is happening at the local level in the network. So we simulate both the dynamic of both nodes as a basal species, but we incorporate them as a subsidy term. So saying that, why this model is an allometric trophic model? Because the biological rate of the species, such as the intrinsic growth rate in primary producer, the metabolic rate, and the maximum consumption rate, the parameter of those values are based on these ecological rules in which or relationship between the biological rate with the body mass of the species, with a scale with a 3/4 value. So doing that, we have this very complex that Leo told me that this a very complex model because we could say simpler, but it's easier in comparison to other models because it's a general model in which we don't need to know very much about the empirical parameter of the model because all the parameter is based on the body side of the species. So the only value that we need are the body side and the biomass of the species. So to obtain this value, we obtain the empirical value from the marine reserve that we have in Chile, ESIM. And this is a marine reserve that had more than 30 years of human exclusion. So our dynamic food web model starts until what we call a without fishing scenario, in which we start to simulate the model until the system reaches a dynamic equilibrium. In that moment, we start to simulate different fishery scenarios, and we simulated until again the system reaches a dynamic equilibrium, and that is what we call it with fishing scenario. So we simulate 10 different fishing scenarios in which we change our Fmax, the exploitation rate value, from 0.1 to 1, which means that we remove the 10% to the 100% of the biomass of each the target species simultaneously at the same time. So now I'm going to show you the result for the intertidal food web in the left and to the subtidal food web in the right. The y-axis is going to represent the biomass change of each species between before and after fisheries. The x-axis represents the species. In this case, I am grouping the species in trophic category just for visualization. And I'm going to show you the result for all the harvested species in these gray boxes and to the non-harvested species in these yellow boxes. And also, I'm going to show you different plots which are organized from the top to the bottom in this different exploitation rate, from the lowest to the highest exploitation rate. And you are going to see bar plots in which you is going to see two types of bars. You can see a green bar, which means that after fisheries, the biomass of the species increases, which is a positive impact, or a red bar, which means that after fisheries, the biomass of the species decreases, which is a negative impact. So in the intertidal food web, the harvested species have response negatively to the impact of fisheries. The biomass of all the harvested species decrease after fisheries, which is quite obvious, right? Because fisheries is directly removing biomass of those species. But the interesting thing is that if we look this first plot, which is the result of our lowest exploitation rate simulation, remember that we explode to all the species with the same exploitation rate. So the interesting thing is that we could observe this difference in the response of the species in front of the same exploitation rate. And we found that these species, which are the producers, the macroalgae, go to extinction with this very low exploitation rate. And what happened with the non-harvested species, the indirect impact of fisheries? We found that the biomass of all the species decreased after fisheries. So this means that fisheries produce a positive impact on non-harvested species, especially on herbivores. And I'm going to explain you the mechanism that explains this in the next slides. And now what happened in the subtidal? We also found that fisheries produce negative effect on the harvested species, which is obvious. But the interesting thing here is that in the subtidal ecosystem, using the same exploitation rate than the intertidal, the species are more sensitive to fisheries than the intertidal. Because why? Because these species have a higher decrease in their biomass after fisheries than the harvested species in the intertidal. And this is a very important result in the case of Chile, because mostly all the landings of fisheries came from subtidal ecosystems. And also we found that the producers are the most sensitive harvested species in the subtidal. And what happened with the non-harvested species? Again, we found that fisheries produce positive impact of the biomass of these non-harvested species. So just looking at this result, we could say that using this multi-species model help us to understand how the impact of fisheries propagates through the network. We could find which are the most sensitive species. For example, we found that the macroalgae are the most sensitive, in which we have the kelps, which are the very important type of species in benthic ecosystem because these species promote biodiversity. But if we think how fisheries work in Chile, we know that kelps now are facing an important problem because they are having a very high intensity of illegal fishing. So this result has been very, very important to fishery management because it's highlighting this message that something is need to do in kelp fisheries to ensure their persistency. And also we can compare how these two benthic ecosystem responds until the same exploitation rate, and we found that the subtidal is more sensitive to fishing than the intertidal food web. And the interesting result that we found, and I would like to stop a little bit here, is that we found that fishing produced positive impact on the non-harvested species. And this is a tricky result that I would like to explain a little bit more, because I'm going to show you how this positive effect could be not so positive. And now here I am showing you the intertidal network. Now I am is disgraded by each species of the network. And you can see how the non-harvested species, that are these species that are into this box, most of all these species have positive impact after fisheries. And why this is happening? Because as you remember in the model, I am showing you that the carrying capacity is the ecosystem carrying capacity. So all basal species compete to reach that carrying capacity. And this means that if by fisheries we remove biomass of all of these basal species in which we have kelps, which are the species with the highest biomass in the ecosystem, we are relaxing the interspecific competition between basal species, and for that reason, these non-harvested species could grow. But also because the fisheries are removing consumers which have a high predation pressure in several other animals in the food web. So these relaxing in the predation pressure are allowing to omnivores and herbivores to grow also. But we cannot forget that this is a deterministic mass balancing model. So just for the fact that we remove biomass of all these harvested species, we are producing an ecological release in the ecosystem. So doing that, because it's a deterministic model and a mass balancing model, we just could find two types of results. The first one is that if we remove biomass of the system, we need to gain biomass. And the only way here is gain biomass with an increase of the biomass of the local species, as we observe here. But we also could observe that, or the other result is that obtain the arrival of new biomass. And a mechanism that could produce the arrival of new biomass is, for example, by invasive species. But this is a mechanism that we are not allowing in our model. And also, on the other hand, we cannot forget that in coastal ecosystems are open system ecosystems. So in which all the species are interacting with other species based on trophic interaction, based in non-trophic interactions, and also are affected by environmental fluctuation and by migration processes. So inspired in this paper of Rolando, and in collaboration with Sergio Rojas and Francisco Cuevas, we want to move toward an open system allometric trophic network model, in which we want to try to incorporate this mechanism. But before to do that, so sorry, this is what we are doing now. But before to do that, we want to be sure that we understand the model well. And doing some exploratory analysis, I know that this model is robust to different parameter values in each parameter of the model. But I know that the half saturation density parameter, which is a parameter of the functional response, the model is quite sensitive. This is the parameter that was harder for me to obtain the parameter values that could produce biomass result similar to the biomass that we could obtain in the species in the marine reserve. So these are the values of my half saturation density, which is B0. Now I am calling it B0. This is the value that I use in my model. And as you can see, they are very different. We have from 50 to this very high number. And why they are so different? Because these values are based in this relationship in which we have several experiments that show that the half saturation density is higher at lower trophic levels than higher trophic levels. For that reason, to top predator in our model have this very low value, and our consumers that consume to a species that are from lower trophic level have these very high B0 values. So I already told you that this was the value that helped me to obtain a biomass value at the end of the simulation that are similar to those biomass that we found in the marine reserve. But now, in collaboration with Francisco and Sergio, we change the parameter value of each category, in which we use random values based on a uniform distribution. And here are the range of the values that we use it. And the only rule is that the values in each category cannot overlap. To maintain this rule that while you are in a higher value, in a higher trophic level, your half saturation density is lower. So we use 1,000 random values. With each one, we run the model, and at the end of the simulation, we compare the biomass of each species between the biomass that I obtained with the value that I use it with the biomass of the species that was obtained with this random value. And if the difference between the biomass of the species that was produced with my B0 values with the random values, if this difference was higher or greater than 5%, we discard this result. So what we found, this are the density plot. We have 6 plots. Each one represents the result for each row in this table. So this B01 represents the result for this first row in the table. And the red line represents the uniform distribution, and the black line represents the probability to that value, this B0 random value, produce similar result to those results that we found with the value that I used in my model. So in general, what we found that we have a wide range of value that could produce similar results to those results that I found with my B0 values. So what this means, and we found that we could obtain this range of value that we could use. So it's a sensitive analysis because we know that this spectra of value in which we can move this parameter to obtain similar value that can replicate the biomass that we can observe in the marine reserve. So using these values, now I am showing you how change the biomass of each species in the food web. The x-axis represent each of our simulation with each B0 value. So simulation 1 represent the simulation with our first random B0 value, and simulation 1000 represent the result with our 1000 random B0 value. And each color represent the different species in the food web. So as you can see, we have some variability, but we can maintain into a range of variability the general proportion of the biomass of the different species. This is the biomass of plankton, which is our most basal species. And all this group here are macroalgae. And here we have our consumers. So it makes sense because in interterrestrial ecosystems, macroalgae have the highest biomass than consumers, and plankton is the species with the highest biomass. So now that we are sure that our model is robust to a wide range of parameter values in this B0 parameter, now we want to move toward this open system ATN model. So this is all that I have prepared for you guys. Thank you so much for listening.
Thank you, Isidora.
I don't know if you have some comments or questions.
Yes. Well, first of all, we
Thank you.
Yes. There is some questions. No, I only see the
Francisco Cuevas.
Francisco Cuevas. No, but he was just appreciating the presentation.
Okay.
Yeah, but Francisco was working with me, so I don't know if Francisco, if I jump something, you can just show up with.
No, I think that everything was pretty clear. I was just clapping my hands.
Okay.
Yeah.
So, so this is a sort of, well, you mentioned that now you will consider, so that is a work in progress you have with the dynamics represented by levy processes.
Right.
Yeah, okay.
Right.
Yeah.
Yeah, this is the thing that we have been discussing with Sergio Navarrete. We haven't discussed it with you, Rolando, yet, but it's a thing that we are starting to do just right now. Like this result we obtained just like yesterday.
Yeah.
So this is our first, we are just recently moving to this.
No, but that's fine. That's quite fine. On the contrary, I think it's very nice to see how to compare both kind of approaches.
Yes.
Closed system approach,
Yeah.
an open system approach.
Yeah.
Well, and I know, as we know also, you are working with Leonardo in another approach to this problem. Certainly.
Yeah, with price switching.
Yes, and this has I think that this have a connection with control problems that were explored yesterday. So with Quiñinao, probably you will have to work together sometime, because that is essential for fisheries. Huh? Yeah.
Yeah. Yes.
Well, okay, so thank you again. I think there is no other comments.
Gerardo is raising the hand.
No, he's
okay.
Yeah. Well,
no, clapping hands.
Yes.
Yes.
Thank you guys for your invitation, and thank you for all of you to listen.
Thank you.
I'm going to stop